So I'm Tobias and I'm the module convener for this module. And we designed this module basically to give the students an experience in one of the really most cutting-edge molecular biology techniques that are out there at the moment. And so the idea is, so the technique is called genome editing. And it's something that was invented about 10 years ago. It received a Nobel Prize last year, a Nobel Prize for chemistry, I think. And it is a technique for introducing targeted changes in essentially any genome, which is really something we couldn't do before this. We could do this for some model organisms, for some very specialized organisms in a very labor-intensive way, but now it's become much easier and a lot more widely available. And so in this practical, we're applying that to one of our lab models, which is baker's yeast. It's a very simple cell that's internally organized like our own cells are, which makes it interesting to work with. You'll know it because it's the same yeast used for baking and brewing. And what we're trying to do is we have two yeast strains. So you can see them here, perhaps. I don't know how easily that's visible. But you can see one of them is white and one of them is red. And the red colored one has a mutation in its genome, which means that one of its metabolic pathways is not working. So one of the things that yeast can normally make, a chemical agent that's called adenine, can't be made in this yeast. And the lesion that causes this is a single nucleotide change in a particular gene, and it's very similar to some hereditary mutations in human genes. Again, this is in a very simple organism, but it's something that's in very similar form is quite relevant across biology, really. And so what the students are trying to do is use this genome editing technology to remove the deleterious mutation, and so take this essentially sick yeast strain and turn it healthy again. Which again is very similar what you would do in a medical context to remove a hereditary mutation from a genome, for example. Although at the moment this isn't applied in humans, mostly for ethical reasons. That technology will probably come in the future. So this is a little taster of that technology, if you want.